Welcome to the Field to Film podcast. So each year, the Arkansas Soybean Promotion Board, they allocate some of their funding to research. So today, we are here with Dr. Burt Bluhm.
So palmer amaranth is one of the most important weed problems facing soybean producers in Arkansas and a lot of the U.S. And it's a very difficult weed to control. There's a lot of unique aspects about its biology that make it very persistent in agricultural environments, but also it quickly evolves resistance to conventional herbicides. And so our goal has been to explore biological control alternatives that specifically target pigweed to develop those into biocontrol products for control. Yeah, I grew up on a farm in rural Mississippi and went into science originally as a botanist and a chemist, and then moved into plant pathology and have been here at the University of Arkansas for about 17 years.
So if, a farmer, they don't have their pigweed under control, what are kind of the effects of that? Talk us through that.
Yeah, pigweed incurs a lot of additional costs as far as weed management in a production system. And in extreme cases, those costs can be very high. In some cases, resistant pigweed can require hand roguing, so that labor input is considerable. And in very extreme cases, there are reports of field abandonment just because the pigweed basically overruns the crop.
And so specifically, for the Arkansas soybean industry, what is your goal for your research?
Yeah, our ultimate goal is to provide Arkansas soybean producers a sustainable, cost-effective alternative to chemical conventional herbicides for pigweed control. And so this could manifest in different ways. This could potentially be a primary means of control for pigweed in the state, but it also potentially could at the very least provide a rotational chemistry, so to speak, so that existing conventional herbicides last longer. So it gives one more kind of wrinkle for pigweed to have to try to evolve around while it's in the process of evolving resistance to all these other herbicides.
So talk us through your methods and techniques used for this project.
Yeah, so this has been ongoing for a number of years, where we initially collected a large number of pathogens of pigweed from all over Arkansas. And some of these pathogens are better suited for commercial control products than others. There's kind of a Goldilocks or an ideal sort of combination of traits in these pathogens that they attack pigweed, ideally kill pigweed, but also have to have other properties that they have to be very specific to pigweed. They can't attack other plants. And so as we worked our way through this large collection, we settled on two groups of pathogens that are they're distinct in their kind of strategies, but also both of them kill pigweed and are very host specific for pigweed. And so now that we've locked in on those, we're working our way through the details as far as optimization. So one of these groups of pathogens is very, very aggressive on pigweed specifically. And so it kills seedlings, it stunts plants, it keeps plants from producing, the pigweed plants that survive the pathogen produce very few seeds. And so that's one kind of strategy of control. And the other strategy for control is the other group produces what looks like a very host selective toxin. That's a case where we're actively working to characterize this toxin and see if it could potentially be used directly as what we would think maybe kind of roughly analogously to a conventional herbicide that targets pigweed. And so that's kind of our two-pronged approach right now.
And so have there been any, challenges or unexpected, findings?
Yeah, it's always taken some twists and turns. There was some groups of these pathogens early on in the process looked very promising, but then as you work through different parameters that they don't sporulate well, so they wouldn't lend themselves to like large-scale commercial production. And so, as we've worked our way through it, some of the elements of the biology we've been surprised about. It looks like both of these groups of fungi are likely undescribed species.
So when you say undescribed species, can you explain that a little bit more?
Yes, these two groups of pathogens probably have not been formally described by taxonomists. And of all of these millions of species, as far as being formally studied and named and put in some sort of like organization, maybe a fraction of the known fungi in the world. And so it's exciting that these fungi that we're looking at probably fall into that undescribed category, where we may be some of the first scientists to be looking at these species in the context of research. And so extremely exciting, also breaking new ground with the work.
What would you say have been the key findings of this?
The concept of this host selective toxin, that as we're still gathering evidence and characterizing what's going on, working our way through it, if this does turn out to be the case, that'll be a very exciting discovery. In thinking of scaling up, statewide level production of a product that can control pigweed, an advantage of having a fungus that produces a host selective toxin is, in the first place, we might be able to utilize that toxin directly as a herbicide. But even if there's some limitation as far as stability or some production limitation, just understanding fundamentally how that toxin works to specifically attack pigweed opens up all kinds of new doors and avenues for research to develop potentially new categories of herbicides that target palmer amaranth, and have a lot more, hopefully, like host selective control.
How would you describe your next steps for this?
For the first group that we're working with that we think would maybe be best applied directly as a living organism, we're looking at different formulation strategies, timing of application, whether this can be done in combination with other more conventional herbicides, some sort of like combinatorial approach. And so we're working our way through the details there of what would be the most effective strategy. For the other group, we're very, very concentrated on trying to understand this toxin and how it kills pigweed. And for both groups, thinking more and more about scale-up sort of questions, where ultimately what we find in the lab has to be translated to field-level applications to be valuable to producers. And so some of the biological properties of these fungi we're investigating and making sure that if there are any roadblocks, that we have ways to work around those, or possibly even through genetics modify the organisms.
Is there anything else that we haven't discussed that is essential for the producer to know?
One is just the incredible gratitude that we have to producers who, through the board, have supported this type of work, and the sustained support and for this sort of work has been really, really helpful.
It's great work that you're doing, and in the end, it's going to have such great benefits on the Arkansas soybean industry. So thank you so much for your time and your continued work on this.
Thank you.
Thanks for listening to the Arkansas Soybean Promotion Board's Soybean Podcast. We hope you'll follow us on Twitter and even more resources at themiraclebean.com.